Welcome, everybody. We're going to go ahead and get started as the panelists roll in, since the initial part will just be introductions. So I don't know about you all, but I have been looking forward to this panel for the entirety of the week. And I want to thank the panelists so much. It's my pleasure to introduce them. Our communications manager, Anahit, thought that instead of reading their accolades, which you all know, we could start with some fun facts to introduce our panel members. So, for example, many of you know Dr. Dario Alessi from University of Dundee, but did you know that while he was visiting a friend in 2003, he was made an honorary chief of a very small village in Cameroon? Welcome, Chief Dario, and thank you for joining the panel. Next up, we have Dr. Andy West from Duke University. Andy had the opportunity to name a new gene. No, he did not name it something fun like Sonic Hedgehog or West is Best, but instead went for PACRG, which is parkin co-regulated gene. On the weekends, he spends his time driving around his John Deere tractor, and that is the only brand name that we will be promoting today. Welcome, Andy.
Thank you.
Next up, we have Dr. Shalini Padmanabhan. We see her when she has her MJFF VP hat on, but did you know that she is also a trained classical Indian dancer since the age of 5? Very impressive. Her favorite activity to de-stress when she's not solving the Parkinson's puzzle is doing jigsaw puzzles. Welcome, Shalini.
Thanks, Spring.
And finally, we have Matt Fell from Merck. He tells us a fun story from the last LRRK2 meeting when he was coming back on the plane and sat behind Superman, Henry Cavill, who he did not recognize until Cavill was whisked away to a secret VIP area of the airport, and Matt was politely ushered down the path of the non-famous people. Matt, you'll always be famous to us.
Yeah, yeah. Thank you.
It's unclear why Superman would need a plane to fly. My first question would be for Dario. Dario, you've done a lot of work on unraveling the mechanisms by which LRRK2 works. The Rab phosphorylation information was incredibly, I think, informative for the field, and implicated multiple different cellular pathways, including intracellular trafficking, lysosomal function, mitochondrial function. Can you comment on, based on now what we've learned in the past, gosh, how long has it been since that came out? What do we know about
8 years.
8 years, yes. What do we know about how LRRK2 contributes to Parkinson's disease from an intracellular perspective?
Yeah, no, I think labs are just the sort of the starting point. I mean, what they get phosphorylated within minutes of LRRK2 becoming activated, and then Parkinson's disease takes 50 years to develop. So I think there's how LRRK2 is connected to PD is truth be told still unknown, but I think there's 3 hypotheses. One is that it induces lysosome dysfunction when you activate LRRK2, and therefore it's an endolysosomal circuit, and if you get damaged lysosomes, you affect mitophagy, autophagy, other important cellular biology, and this could lead to loss of dopaminergic neurons. The second idea is that you have loss of cilia in rare cholinergic neurons in the dorsal striatum that are critical for sensing sonic hedgehog from the dopaminergic neurons and producing GDNF to provide a survival signal. And I think there's an increasing amount of evidence that suggests that numerous PD genes, including LRRK2, do this. And I think that the third mechanism is GBA. There's some evidence, increasing evidence, that LRRK2 activates and decreases GBA activity. And this could actually be due to lowering the levels of BMP that are critical regulators of GBA. So those are the 3 pathways that I'm sure the other panelists will have other pathways to suggest that are linked to Parkinson's.
Wonderful. Thanks, Dario. Andy, anything to add to that?
I would add, I don't know if it's a pathway so much as a process, but we've been drug, I suppose, kicking and screaming into neuroinflammation and inflammatory pathways. LRRK2, entered the realm of Parkinson's with some baggage and links with other inflammatory diseases, mycobacteria infection, some of these other, disorders where inflammation is prominent. And, I suppose in complex disease, there's examples where there's cell autonomous effects that are also later on modified by, cell non-autonomous effects. And I suppose I think about LRRK2 in inflammation in the latter category, where maybe in established disease, there could be a different emphasis than the initial origins of disease. And it's complex, There's not likely to be oh, I shouldn't influence the poll.
It's okay. You can influence the poll.
Multiple pathways.
Well, multiple pathways, I think is kind of the easy way to go. But is there a way, can anyone think of any particular experiment that might suggest a specific pathway? Is there an experiment that you could see and go, definitely it's the lysosomes that are involved, but sure, all of these other things are happening, or yes, definitely it's inflammatory. I think it's difficult to separate them, but
I don't know, Spring. I always feel that the best experiment is done in a human. And I feel like, the disease is so heterogeneous that selecting, one particular model is not going to be useful. So the way we have been thinking about it is, how do we elevate the best biomarkers, pathway-based biomarkers, into cohorts such as, PPMI, the Parkinson's Progression Markers Initiative, and try to look at now, what's really regulated in those LRRK2 individuals, right? I think that's an easy experiment to do, but I think our assays are not there yet for many of those for the pathways. And it would be interesting also to hear from the other panelists, because now we have data on the synuclein seed amplification assay, and we can touch on this later as well, try to understand, are there multiple mechanisms that are being regulated by LRRK2 and that are leading to different outcomes in different individuals? I think those are all interesting questions, and I think easy to kind of come up with hypotheses, but I think more difficult experimentally because we're so behind on the assays.
yeah, totally agree on needing more assays to measure these various different biological pathways. And certainly that's I think synuclein seed assay, I'm hoping, is the first domino leading to multiple of the other ones, and surely we're working on them as well. You mentioned synuclein, Dario mentioned GBA. Let's talk about some of the convergences, because I think that might give us a clue as to how, assuming there are shared pathways for various different, GBA, PD, LRRK2 PD, synuclein PD, and we shouldn't assume, of course. Dario, you mentioned GBA PD, and Andy, you and I had discussed this before. There are a small subset of patients that have both GBA and LRRK2 mutations. And it was certainly not intuitive to me when I first looked at that it suggests that, at least the cognitive decline is no worse in the GBA plus LRRK2 patients than it is in the LRRK2. It's worse in the GBA mutation carriers alone. Now, I know that there's not a lot of power because there aren't that many patients in that study, but any thoughts, Dario, on that?
No, I find that one of the most puzzling. I believe there's the science is obviously humans. It has to be right, I think, but it doesn't fit in with our current understanding. So I clearly, it's a very important thing that we need to think about, and it could mean that there's something strange here. Normally, when you have two disease-causing mutations and you combine them, you exacerbate the disease. You don't make it slightly better. So it's a bamboozling observation that I can't think of an explanation for.
Andy?
Well, I see Shalini smiling, because I love thinking about these things. So I agree, Dario, with everything you said. Shalini, I agree with you said in terms of top-down approach. I think we start with the patients and hope that our models can replicate something that we see happening. And the only thing I would take issue with, Dario, that you said is assuming that GBA is the cause of Parkinson's. Because if we assume that GBA is critical for progression, in idiopathic synuclein disease, and LRRK2 is important for risk and initial development in G20 carriers. They may not exactly align, especially in the LRRK2 carriers that are synuclein negative. But even in LRRK2, that's showing up in seeded assays, my impression in looking at these brains, they're pretty rarefied for synuclein pathology. So GBA can't put its evil hooks in progression because it's not quite the same disease. You get some of the same phenotypes, sort of, but there's a different path. And I think you can set up models that are biased for one way or another, but I think one thing we have difficulty unraveling is risk versus progression. And LRRK2, I think, is the one of the most potent, G20 and the other mutations are some of the most potent risk factors. And I think the GBA variants are some of the most potent factors we know about that modify progression. And so the difference, in intersection in these patients might be what touched off the disease in the first place. And certainly I can tell you, in working with synuclein models with LRRK2 for the last 15 years, it has been absolutely hit or miss. We see some interactions with LRRK in different synuclein endpoints, and then it doesn't touch others. And it's been really frustrating, trying to get them to fit in the same pathways. And it's something we still, struggle with, I think.
Thanks, Andy. I do want to talk about synuclein, and that's been brought up as well. Shalini brought it up. Of course, the relationship between, you can't really talk about Parkinson's disease without talking about synuclein. And the relationship between LRRK2 and synuclein has not been a simple one, Andy, as you just mentioned, right? Not all of the synuclein, or not all the LRRK2 patients manifest with synuclein pathology. Even in the seed assay, we don't necessarily see synuclein. In the preclinical models, we don't, sometimes there's a relationship, sometimes there's not. It's not really consistent. Shalini, do you want to comment on that?
I'm curious to also hear what Matt thinks, kind of coming from the industry side of things. I think it's been challenging to use synuclein as a model for LRRK2 therapies, and specifically kinase inhibitor therapies. Of course, there are other modalities now that we can also talk about. But I don't think the kinase inhibitors have been successfully demonstrated efficacy in synuclein models, and that hasn't deterred companies, right? So I feel like that wasn't something that prevented them from going into the clinic. And so I feel this is an interesting question that I think remains to be answered. And I think the connection between LRRK2 and synuclein, or LRRK2 and other aggregate proteins, and I'm talking about tau now, where there's been several instances, both from postmortem brain tissues and also from preclinical models, where LRRK2 does seem to impact tau. And so it's interesting, because if we think about the disease as a synuclein-based disease or a non-synuclein-based disease, and now with all of the new staging, conversations that are happening, it's possible that you think about, the patient, population differently for these trials, right? So I think it's important to think about that and talk about that, but I don't think there's been a clear-cut connection between kinase activity and synuclein pathology in preclinical work. Now, that's different when you talk about LRRK2 levels, because I think that's where, when, a clear reduction in LRRK2 levels has shown to be, has shown to have different phenotypes when you compare it to kinase inhibitors. And this, I think, comes from Andy's work and Virginia Lee's work and things like that. So curious to hear what Andy and Matt think about these data.
Matt, can you add your translational perspective? And we'll talk about the therapies at the end, but sticking with the theme here.
Yeah, no, I agree with Shalini. I think, it's been very difficult preclinically to demonstrate impact of kinase inhibitors on synuclein pathology. And internally, we've done a lot of studies with PFF models looking at, seeding G2019S mice at early ages, really didn't see any exacerbation of pathology there, didn't see any modulation via a kinase inhibitor. Cheryl Lyons in my group actually, did a really nice study where she ultimately showed that the G2019S mutation carrying mice have, deficits in the GBA pathway with elevations in glucosylspheric sphingosine. And we ended up seeding those animals once they had that elevated level, and we still didn't really see a difference in the change in alpha-synuclein pathology with or without sort of the treatment. So although we did actually see in some of our studies where we didn't do the seeding, we did see modulation of the the lipid levels via LRRK2 kinase inhibitor. So I think that's an area that's been difficult. But then we actually had a study with Michael Henderson where he looked in the G2019S and suited with the tau, and in the mutation carriers, not the wild type, he did see a modulation of that tau phenotype. So it's been it's been quite challenging. I think from a discovery perspective, I think we look at it as though that there are limitations to animal models, right? And so depending on what your company's philosophy is and how you target the disease, do you understand biomarkers and do you understand the hypothesis that you're trying to go after? How much weight you put therein to sort of preclinical animal models. But it's always nice if you have that feeling that you are modulating an integral pathway of the pathology, but it is rarely that simple.
Thanks, Matt. And I think that's a really important point as well with these preclinical models from a therapeutic development perspective. Are these clear go, no-go points, or is it a nice to have? And I think us included, we do them, but it would not be a clear go, no-go because the preclinical models are flawed, right? Dario, Andy, any comments on synuclein and
I would just I don't understand how synuclein and LRRK2 interact. I find the literature is very confusing as has been discussed. But I think the only point I think that I would like to make is that for the clinical trials that are taking place, I think it would be very important to to work out whether the response or any response to the therapy, how it's affected by the presence or absence of alpha-synuclein in the seeding assay, because one might predict that the individuals without alpha-synuclein might respond better to LRRK2 inhibitor therapy, or it'd be interesting to test that at least. So I don't know if that's being done, but if it isn't, I would hope it's being done.
Yeah, I'm not sure. Anybody know if that's actually being done in any of the active trials? And we'll discuss the trials at the end.
I don't know if it's being incorporated, but I mean, I do think you're starting to get to a point where you have seeding assays that may enable you to select patients who are at least alpha-synuclein positive. But then if you can start thinking about some of the other PET traces that are coming online for things like tau that you could potentially leverage, and I know Shalini's group has been doing a lot to image patients with some of the tau imaging agents. I mean, I think that might get to some of sort of Dario's questioning, but then to do that subgroup analysis, you need to have a significant number of patients, right? It has to be powered appropriately, and then we start getting into this challenging space.
Yep, makes sense. So moving off of synuclein, tau, and GBA and onto other genes that are connected to PD. Dario, I saw you had a publication alongside Scott Small, who's going to be on the VPS35 panel next week, or in a couple of weeks. What do you think about the convergence there on the endolysosomal pathway? That's also interesting in its own right.
I think there's a specific form of lysosomal stress that we don't understand what it is, but that's relevant for Parkinson's disease. And it's probably, I think the VPS35 mutations affect cargo transport in the endosomes, and therefore the endosomes become lysosomes eventually. And I think if you have the wrong cargo in the endosomes, you probably end up with some protein imbalance in the lysosomes that causes some sort of physiological stress, and this leads to the active recruitment of LRRK2 to the lysosomes and the phosphorylase and the Behrouz proteins, and then their effects as binders. It's quite a complicated pathway, but I think if we could really understand what the stress was and how to study it and measure it in people, and then in drug screens to look for things that counteract it. I mean, one way to do it is you could use the LRRK2 recruitment or the LRRK2 activity, the Rab phosphorylations on the lysosomes, as a readout to look for things that counteract that. But that's a sort of interesting question that would be worth investigating, I think.
Any other comments from the panelists on VPS35? It's not a very heavily studied one, certainly not compared to LRRK2.
It's the one thing that activates the LRRK2 the most that we know of. So in many systems, and also in blood cells from patients, the effect you see in RAB10 phosphorylation is much higher in the whole cell extract with VPS35 than with LRRK2 pathogenic mutations. And if you isolate the lysosomes and then look at the RAB10 phosphorylation, then you see like up to 64-fold increases in the RAB phosphorylation associated. So it's really having very profound effects. So although it's rare and understudied, it's probably very interesting to understand how this is working.
Agreed. I will.
And I
be
look forward
bringing
to
this
next
up.
week's call. I'll be sure to join you.
We'll discuss it from the other side next time.
I was just going to say, Spring, I mean, to Dario, I mean, to your point that it's sort of understudied because the patient numbers are so small, but has there been any pathology on those patients to see how that may line up with with the LRRK2 mutation carriers, given that it is sort of a hyperactivating in effect?
I think Darren Moore may have done some work. I don't know with so he's certainly done very beautiful work with mouse models and knockout and disease models. But whether this has been done in humans, I should know, but I haven't seen it, so maybe there hasn't been done.
Yeah.
So.
I'm not sure, Matt. I'm not sure if there's like postmortem brain pathology work that's done, but I just wanted to kind of just quickly plug in a study that we initiated spring, kind of based on a mini workshop that we had last year on VPS35, and we felt it was important to understand this intersection between VPS35, LRRK2, and just pathology in Parkinson's disease. So we actually are working with Alexander Zimprich to now recruit like 20 or 30 individuals with VPS35 mutations, and working closely with Dario's group and Christine Klein from GP2 to collect CSF and PBMCs to study kind of lysosomal dysregulation in the periphery, but also look at seed amplification assay to see if it's the same kind of frequencies that we see in LRRK2 individuals. So we'll be happy to report back in a year or two based on what.
There's lots of patients in Austria. I don't know why, but Alexander Zimprich has about 30 patients that he can access.
Very nice. That should definitely be very interesting. Thanks, Shalini, for bringing that up. The other pathway that hasn't been talked about much is the pink parkin pathway and mitochondria. So I do find it interesting. It often gets left out when talking about LRRK2, even though we've seen mitochondrial deficits. I know when I was working with Heather Melrose at Mayo and Matt Farah's lab, she created these G2019S knock-in mice, and we could very clearly see this change in mitochondrial quality control. The morphology was very different. And since then, it's been reported by many different groups. Dario Alessi at Dundee has also more recently reported some nice data on that front. I'm curious what you all think about the mitochondrial side of things. Dario, maybe starting with you.
Yeah, no, well, my close colleague, Miritor Makic, has done some work on this, where he's crossed the pink and the pink pathogenic and the LRRK2 pathogenic mice together. And, surprisingly, knocking out pink doesn't affect LRRK2 activity, even with the pathogenic mutation, and vice versa. Introducing LRRK2 into the pink knockout model doesn't seem to affect the biology or that one's measuring. And even if you purify mitochondria from the LRRK2 pathogenic mice or the VPS35 pathogenic mice, surprisingly, the 900 proteins you see in the mitochondrial aren't really affected. So I'm sure there must be some interplay here. But to me, the way we're interpreting this result is that you'll need different therapies for people with pink and parkin mutations compared to LRRK2. For pink and parkin, you probably need boosters of mitophagy or autophagy, whereas for LRRK2, it's just, so yeah. I mean, maybe others have other stories, but yeah.
Yeah, interesting. So there are, non-pink parkin mediated mitophagy pathways as well. And I wonder, has anybody looked to see if LRRK2, because LRRK2 does seem to impact mitophagy.
Yes,
and I don't think the mechanism by which it does.
I think for me, I interpret this with the dysfunctional lysosomes. So if you have LRRK2, you sort of the lysosomes are slightly dysfunctional, and therefore that automatically will lead to reduced mitophagy or autophagy simply because of the lysosomes. So
So some kind of a
it's
feedback
a bit
mechanism.
indirect. It's a bit indirect rather than directly activating the triggering of mitophagy that pink does. It's more the end stage. The lysosomes maybe are a bit less efficient at chewing up the damaged mitochondria, but that's just a hypothesis, and I'm not sure. We have no data for that.
Shalini, any thoughts on that side?
I don't actually, and I kind of tend to agree with what Dario says. I feel like it's an indirect effect that's probably downstream of, you know, something that's happening at the lysosome. But I think we've seen effects beyond mitophagy, right? Spring, I feel like, you know, respiration and other things have also been shown to be impacted in LRRK2 mutation models. And so, but I don't think we've ever seen kind of a direct action of LRRK2 on mitochondria. I mean, there's lots of interesting kind of tidbits of information around mitophagy and mitochondrial DNA damage, you know, from Laurie Sanders' lab, but I don't think it's a direct connection. So I'm just curious also what Andy thinks. Andy, have you done anything mitochondria and anything here?
No, I would just echo, I think, what was said. I mean, you know, if one considers these relatively distinct disorders with overlapping, you know, I suppose cellular sort of intersections, you know, I suppose I started in, you know, working with parkin in Parkinson's disease, and we had hoped, you know, the gene and the pink-1 axis would survive in GWAS, you know, and that as we increase the numbers, we'll start to see signals that sort of flesh it out a little bit, but they stayed confined to early onset PD that, you know, if LRRK2 doesn't have a lot of synuclein, parkin pink-1 have even less, right? So, you know, and I guess the world to me fits together if you think of LRRK2 as was mentioned, sort of upstream, you know, in terms of these problems that can phenocopy some aspects of a downstream, you know, profounder mitochondrial deficit that's a little bit more focal. And you know, that could be one of the key ways that dopamine cells die, is maybe not, you know, from the inclusion they show, but maybe from, you know, the mitochondrial destruction. And there's more than one ways to get there, and parkin pink-1 axis might be one of those in those patients. Ultimately, you know, I don't know how you're going to figure that out in models. You know, you really have to have an ability to, you know, go in and be able to manipulate, you know, some of those damage responses, I suppose.
Thanks, Andy. I think that brings us all the way back to what Shalini mentioned a bit ago, which is measuring multiple things. So I do wonder, and I know even if we think it's downstream, as Andy just mentioned, that doesn't necessarily mean it may not be important. It may be that mitochondrial dysfunction is kind of the furthest downstream thing that then leads to the neurodegeneration that we see. So I hope that if I can put a plug in here, as you all know, we work on mitochondria. So at least measuring those, that pathway would be interesting, at least getting that data and not just making the assumption that it's downstream, it doesn't matter kind of thing. Okay, moving off of this, did I miss any genes that anybody wanted to talk about that converge with LRRK2 or don't converge with LRRK2 or are surprising like GBA in the patients?
I mean, maybe I could just mention very briefly that the work of Matt Farrer has identified a new variant of LRRK32. It's very rare. It's only found, I believe, in around 13 people so far. But this variant seems to activate LRRK2 more potently than the wild type variant, and it binds to a region called site 1 on the armadillo domain. So you know, that's quite interesting. Yeah.
Thanks, Dario. I saw that as well. Definitely very interesting. We'll also link a lot of these studies that we're talking about in the video, so if people want to go back and have a look at the studies, they can. Okay, moving on to actually, one more thing before we move off of this is the cell types, and maybe we can just very briefly address this. Neurons, glia, other cell types. Andy, maybe you can comment in on that since you've done some work on non-neuronal cells.
Yeah, I mean, I think, when the sequencing databases and single cell, profiles were coming out, I think we had a bit of a surprise with how different one cell to the next can be with respect to LRRK2. But the other surprise we had is how different, different mammals can be, even, from human to mice. That we did an early study where we thought we were going to get in that trouble because the promoter is completely different from, human to mice. The non-coding regions aren't very conserved at all, whereas the amino acid sequence is highly conserved. So we need LRRK2 function, but it seems different species have put the protein in different types of cells. One thing that seems to be conserved, as far as we can find all the way down, is putting LRRK2 in macrophage-like cells. Whether it's a neutrophil or a granulocyte. And we've spent a lot of time working with those cells because that's where we can sort of purify the protein, we can stain for it, we see its activity profiles. But then when we go to other cell types where we know LRRK2 is probably having effect, like a dopaminergic neuron, we've struggled to be able to see, its footprints in those types of cells. So I think, as we get better in, being able to resolve LRRK2 activity, whether it be through RABs or other manifestations, I think the next level is figuring out, expression, versus activity. How much do they actually correlate? People have done a lot with LRRK2 mRNA and looking at different databases and seeing effects of GWAS, but we haven't so far seen a strong effect on activity outputs. It seems that LRRK2 expression may not be as important as other regulatory factors. Some of them, I think Dario already mentioned in terms of other genes and mutations. So I think that's the next level. But we've had a hard time, figuring out where. It's not a very abundant gene. I mean, in single cell databases, it usually hangs out in the low parts per million, with the exception of maybe a few cells, and it's difficult to work with those proteins.
Matt, can I get your perspective since you're director of the neuroimmunology group?
Yeah, no, I mean, it's been fascinating, right? As Andy says, it's been a hard protein to work with. And I think the assumption was because it's Parkinson's disease, well, we've got to look at neurons, right? But over time, we've really been forced to move away from that. And so, I think the role of LRRK2 and microglia is really interesting. But one step beyond that is it centrally mediated, right? We assume that it is, but it actually there's a lot of LRRK2 in the periphery, and is LRRK2, activation there, contributing to the pathogenesis of the disease. I mean, these are really tough questions to answer, unfortunately, but it's one that I think is one that fascinates people about LRRK2 and why we're also interested in it.
Yep, and we do have a poll out on that one too, and it's looks like the majority are. I thought about not including the multiple pathways and multiple cell types because it seems like that's the logical, but that's winning. But neurons are leading microglia. Dario and Shalini, any thoughts on this front?
I don't have anything further to add, except in an ideal world, you would have an animal model of Parkinson's disease, and you could give it, brain penetrant and a non-brain penetrant inhibitor to see which one works. And then that might help answer that question, but I don't think we can do this experiment. So,
Agreed.
Something to add, and something that I've been seeing recently, and also curious to hear what the other panelists think, is clearly in neurons, there is the kinase activity seems to be important, at least reverses some of the PD phenotypes. In many of the immune assays or the immune models, I feel like clearly the kinase activity is not, playing as much of a role, and it's more kind of expression related, because as soon as you change the expression, you see changes, but kinase inhibitors don't do anything. So I'm very curious, what the panelists think about like expression versus kinase activity in immune cells versus neurons respectively. And then And I'm very, very intrigued by the intersection of this lysosome and immune pathways in immune cells. So I feel like there's something to do with lysosomal dysregulation in immune cells that is most relevant to Parkinson's disease. But I have not seen like a killer experiment done here, and happy to entertain, any ideas in this space.
I think as Dario was saying, if you could have specific knockdown or knockout, and certainly if we had a nice model of Parkinson's disease, which I think we'll have a therapeutic before we have a nice model of Parkinson's.
Obviously, then you could
Yeah.
I could add the brain penetrant LRRK2 inhibitor and the non-brain penetrant and the PROTAC brain penetrant and the non-PROTAC. And then you could really answer, Shalini's questions. I know the work of Matt Gutierrez and others does suggest that there's, independent, which isn't surprising because it has a GTPase domain and other domains. And yeah, I think we will, but I think most of our knowledge at the moment comes from the kinase dependent functions, and I think all the PD data points towards activation of the kinase being critical. But obviously, sure, I mean, I'm sure that it's very important to explore non-kinase dependent effects as well.
And so we're naturally kind of going into the next, the translational component of the panel, which is, their kinase inhibitors. I think to what I have seen, these are really the 3 active clinical trials that are going on for LRRK2. If you know any other ones, you can see my screen, I'm assuming. If you know any other ones, please speak up. But there's the small molecule kinase inhibitors that were developed by Denali that are being taken forward by Denali Biogen. That is the phase 2b, as far as I know. The LUMA's continuing. They discontinued the Lighthouse, but they added the LRRK2 PD patients into the phase 2. So hopefully we should have some data on that at some point. I think that'll be the first real inpatient readout of a LRRK2 approach. And we have the ASOs, Ionis and Biogen. That will be, I believe that's intrathecal, still in phase 1. So we'll see. And then the PROTAC degraders that we mentioned, I know, Dario, you've had a couple of papers on that. This group, Arvinas, just announced a couple of weeks ago that they dosed their first human patient. So that's also in phase 1. Shalini, you've brought up a couple of times now.
I think there's one more.
Yeah, Neuron 23. Yeah.
Oh,
Neuron 23, yeah.
I knew that. Okay, right. Apologies to those guys. So I'll add that in. Thanks, Shalini and Dario. So any comments on this expression versus kinase activity and expression changes with ASOs versus the PROTAC degraders? Dario, maybe you can get us started.
Yeah, no, I think the LRRK2 PROTAC is very interesting because it's catalytic. So the degrader will degrade one molecule and then it's free once to degrade more. So you can actually use it at quite low concentrations compared to a conventional inhibitor where you need to inhibit 70 or 80% of the enzyme. So I think. Yeah, I think the ASO, you have to do this intrathecal injection, and I don't know how whether patients enjoy that, and whether this goes in uniformly into every region of the brain. But a small molecule might go in, to the deeper regions of the brain. And if you listen to the Arvinas seminar that's online that I listened to a few days ago, they claim that the PROTAC goes deep into the deepest regions of the brain and reduces LRRK2 expression homogeneously in these regions. So it's a very interesting development. And we were just chatting before when we worked on the LRRK2 PROTACs, we still haven't got a grant to work on this because everybody told us that PROTACs won't work for neurodegeneration. So every time we wrote a grant, they got rejected because of that. And every time we talked to anyone in a pharmaceutical company, they basically said the same thing. So, I'm really pleased to see that, this mindset isn't the case. And I think there's many interesting sort of undruggable targets that could be considered in the space of neurodegeneration with this PROTAC technology.
Matt, can I get your industry perspective here? Obviously, you guys worked on LRRK2 inhibitors, the kinase inhibitors. I don't know if you've worked on any of the other two approaches, but what are your thoughts on this front?
I mean, yeah, I can't sort of disclose what we haven't worked on, but yeah, I mean, I'm
Give us
really,
all the details now.
yeah, I'm really intrigued by the approach, to be quite honest. I mean, to Dario, your point, right? I mean, you can say something won't work and you'll be right 99.9% of the time, right? So it's really easy to do that, and it's hard to actually back the things that will work, and it takes perseverance and a lot of courage to do that. So, you know, I'm really intrigued to see how these might work. I mean, you know, certainly some of the data looks really interesting, and that would be great if that can, you know, if that can offer a differentiated way of targeting from a kinase inhibitor, and then potentially be differentiated from a from a safety perspective, and then that could be really important.
Andy and Shalini, any thoughts?
You want to go, Shalini?
You go first, and then I'll follow.
Well, I think one of the original questions was intersection of kinase inhibition versus reduction of protein. And you know, one of the key findings, I think, from the Denali phase 1 study was that they could reduce total LRRK2 protein in CSF by about 50% in many of their patients, or many of the participants. And I think it's not yet been settled whether that's because of destruction of the protein itself from cellular sources that contribute to the CSF, or because, you know, they've reduced exocytosis of LRRK2 into biofluids. We sort of can make pathways for either scenario. So you know, Warren Hurst and I would go on endlessly about why it seems some types of LRRK2 inhibitors destroy LRRK2 protein in some types of cells and not others. And it can be very, I know, Matt, I think you've seen this before too. And
yeah,
you know, I don't think we have an idea yet, but I think it's going to be, you know, important as to the sort of fundamental biology of LRRK2. But in the end, maybe the inhibitors aren't so different from the degraders when it comes to biomarkers, you know, that can be tracked and recorded in terms of seeing the protein going down. And then it's a question of, you know, what's your target? You know, is 50% enough in the brain? It seems low to me. It doesn't seem like enough, because we've never really seen any effects of the heterozygous knockouts. You know, and I know Ionis has gone over this forever with trying to figure out how much of a target do they need to reduce. And I know there's steadfast, you know, people holding the line that even 70–80% is not enough for some of these aggregation, you know, proteins. So I think it'll be interesting to see how LRRK2 evolves. I think the Denali study sets a bar for the inhibition that they can find. And I am certain that other therapeutics in the future, you know, there's space to improve on how much inhibition. And then again, I think Dario mentioned it might not be desirable to completely destroy it all. You know, maybe some remaining, whether it's in the lung or kidney, is part of, you know, a successful strategy. And Matt, your lung data is quite compelling, that it you know, you have to be careful targeting this kinase. So yeah.
No, I would agree. I would agree with you. I mean, we've had a lot of experience looking at LRRK2 protein levels in response to kinase inhibitors, and it's really cell type specific. You know, certain cells, you see it in vitro very quickly, you'll see a reduction over sort of 18 hours. Other times you go in vivo and you don't see it in even lung tissue and things like that. And it's really quite difficult. And then you layer on top of that, like we've already been discussing, about what cell type is important, what's the location of it, and then, you know, is that the cell type that you need to be reducing LRRK2 protein levels in? You know, I think it's just, you know, it's another layer of complexity, and the first iterations of therapeutics, they'll target certain hypotheses, but rarely is it the first entrant that sort of gets it right, and so hopefully there'll be learnings that come out of the ongoing clinical trials. We can sort of refine the hypotheses, but yeah, our data would sort of suggest that certainly getting to those very high levels of target engagement, at least in non-human primate, may not be something that you want to do. Is that what you need for efficacy? That's a question we don't know, right? And that's the sort of the dilemma here is 50% enough? Is bringing that kinase activity back down to wild type levels? Now, these are patients who've had that for lifelong versus an 18-month clinical trial or whatever it will be. So it's complex.
And so thinking about, those two questions, if we can just separate them for a moment, obviously we do have to think about efficacy and safety, but let's just separate them for a moment. From an efficacy perspective, we want to, am I getting the sense from the panel that degrading would be maybe better than inhibiting? Maybe not.
I think it gets complicated because all the inhibitors that have been tried so far are type 1 compounds that theoretically trap the enzyme in this pathogenic conformation. So although you inhibit the pathway, you may get some effects from the pathogenic conformation or destabilizing the enzyme because it goes into this active conformation. So that could cause complications. So if the degrader does work, theoretically, it's not a bad approach because you're dialing down the enzyme, and you can maybe dial it down more easily by using the right dose of the compound than you can with an inhibitor that may have different effects depending on the time you've taken the pill. So it just needs to be, it remains to be seen, but it's not, if you think about it, the degrader could end up being a very good approach.
I mean, and to Shalini's point too, though, right, versus kinase activity versus expression, the profiles could be quite different in vivo. So, I mean, in theory, the way I look at it, a small molecule kinase inhibitor will inhibit the kinase-dependent functions of LRRK2. But if you clear out the protein, and assuming that protein has other scaffolding functions or non-kinase related activities, then by removing that protein, you're going to impact those functions in addition to kinase activity. So, it's
yeah, no, that could happen as well. Yeah.
Yeah.
Yep. No easy, simple answer to the complex question. Although I think I do find the exercise helpful to sometimes step back and say, if I were a patient with LRRK2 PD, which of these three and I had the option to take one of these three, which of these three would I pick? And you can't say all three may work or there are some benefits to one or the other. But in the long run, we shall see with the clinical trial. So I'm excited to get that data. From an on-target liability perspective, I know, Dario, you mentioned, and of course, this is, I think anybody would pick taking a pill over having an intrathecal injection, but having it more directed there, what do you think about the, potential effects on the lungs or other peripheral organs that way?
You talked to me, Spring, yes. Yeah. No.
Oh, sorry, yes.
Yeah, sorry, apologies. Well, no, I think, obviously Merck's data is extremely compelling. I mean, and that it's clearly, obviously, a very high doses, I believe, that Merck are using here that probably kills the pathway 100%. That you are getting very severe, irreversible lung pathology, and yeah, I think maybe Matt can comment, but from my end, I'm not a pathologist, I'm a biochemist, but from my understanding is that Merck are doing additional, very sophisticated cuts and pathology of the lungs on the primate models that maybe other companies aren't doing. And I would think it'd be very critical that every company does these additional tests. I believe Merck are used to sort of smoking diseases, so they've really optimized this approach. And maybe neurology companies aren't at the same level. Maybe this is a bit controversial now, but I think I would like to see the same pathology being done in the non-human primate models. across all the data, the trials, the preclinical trials.
Yeah, I'm certainly not a pathologist. I play one on webinars but certainly the approach that I've been led, it's been discussed to me is that the approach that we took is really from our inhalation programs, where you're trying to capture, compounds, activities that are deep within the lungs. And so it is a more broader sectioning profile that is conducted that may not be sort of routinely done. Certainly actually isn't routinely done on non-inhaled programs internally, or at least wasn't. So, that I think probably is a critical factor in assessing that pathology. And certainly, to Dario's point, the molecules that we were able to take in were very highly potent and selective and well tolerated, and so that enables you to dose up to those higher sort of exposure multiples that you're targeting. Now, they are high, but they're not super pharmacological in the most part. They are at the higher end of that occupancy curve, which is where you're starting to see some of these effects. But so.
Okay, one last very important question, which is, do we think these LRRK2 centric therapies will work for some PD patients? And if we do we think that will be a large subset or a small subset of the idiopathic Parkinson's group? And Shalini, maybe you can get us started on this.
My favorite question this is, Spring. I honestly, yeah, I mean, I feel like we've had so many discussions around this that I feel it's important. And I also feel it's going to be very challenging to find de novo, patients with LRRK2 mutations to run clinical trials, right? So I feel like it's important to start thinking about the recruitment strategy early on. And I honestly, I don't think two people with LRRK2 mutations are the same either. I never thought it before the synuclein seed amplification assay, and now I'm even more convinced that even the LRRK2 G2019S population is not a homogeneous population. So it'll be interesting to see how we kind of dissect that data from the Denali trial. I'm sure they're thinking of all sorts of ways to analyze the data. But in terms of the idiopathic PD population, I feel Neuron 23 has an interesting angle there. So they have been looking at LRRK2 biomarker signatures and genetic and clinical signatures, and trying to see what subset of the idiopathic PD pool, look like LRRK2 mutation carriers. And according to them, they are able to find 23%. So they have a mechanism to now recruit, a subset of those idiopathic PD individuals into their trials. So it'll be very interesting to see, what that looks like. I mean, we know for at least from Mark Cooksen's study and the genetic study, that there is a common variant in LRRK2 that's, I think that's present in about 13% of the idiopathic PD population. And he's shown very nicely in microglia that there is an association with levels. So there is increased levels in these individuals. So I would be very curious to see, if Biogen and now Arvinas kind of use that information, in their trials while recruiting patients. So I think people have to start now, collecting this data, gathering this information as they put together their trials. But I think there are strategies out there. I think people are thinking about expanding beyond just LRRK2 mutation carriers. But these are the studies that come to my mind, when you raise that question.
Thanks, Shalini. Certainly the Biogen study is including idiopathic patients as well. I would be curious how much of that kind of sub-stratification, or at least collection of data is being done on that front. That would be nice to know. Thoughts from the rest of the panel on the same question?
Yeah, I was slightly disappointed when I heard that the Biogen had stopped the Lighthouse trial that was just with the LRRK2, people. You say there's few patients, but there's probably several 100,000 people in the world, even more, who have the LRRK2 mutation. And I'm getting more and more emails from healthy people with the G2019S mutation, and they're all asking me, can they take the LRRK2 inhibitor to prevent them from getting Parkinson's disease? So, I think it would be nice to do the trial just with those groups of people and the idiopathic, and I think it's, I think that it will work. But the question is, when do you need to start the treatment? And the answer might be you need to start it, you know, at the prodromal phase of the disease as well, which shouldn't be forgotten about, and how we do these trials, how we pay for them, you know, are big questions.
Yeah, I mean, we're sort of at the infancy of disease modification trials for Parkinson's disease, right? And you see just from the amyloid A beta field, how many iterations has it taken, and patient segmentation and the biomarkers there to get to that cohort where you can generate efficacy appropriately. I think, you know, for LRRK2, you know, the idea of having a genetically defined cohort is really attractive because you can sort of test your mechanism in that disease population. But then when you start layering into other factors such as what stage of disease do you want to intercede in, and then building a cohort of patients within a reasonable time period that, you know, the pharma companies have to work in, it becomes quite challenging. So I'm interested to see how, like, you know, the Neuron 23 approach goes, right, where you find patients who are building off of the work that's been done like Dario to identify idiopathic patients but have a LRRK2 type signature, and will they give you the sort of the best possibility of detecting your efficacy signal?
In the 1 minute we have left, can I just ask one question that was in from the chat, which is, do we know, this came from Warren Hurst, do we know what pool of LRRK2 in a cell is targeted by the PROTAC and how it differs across cell types? Anybody have the answer to that? I don't know.
I don't think
Nobody knows. All right. Well, thank you so much. We're right at 1 o'clock. There are many questions that we didn't have time to ask, but I thank you all so much for being here and for this very informative discussion. And I look forward to bringing some of these up when we discuss the other genes that are involved in Parkinson's disease as well. Thank you, everybody.
Great.
Thank you.
Thank you.
Thanks
Bye-bye.
for organizing, Spring.